Hello, everyone. Welcome to today's live broadcast, High Content Imaging and Analysis for Drug Discovery, Phenotypic Assays, Instrumentation, and Software. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. Thermo Fisher Scientific Incorporation is the world leader in serving science, with revenues of $18 billion and more than 55,000 employees globally. Their mission is to enable their customers to make the world healthier, cleaner, and safer. They help their customers accelerate life sciences research, solve complex analytical challenges, improve patient diagnostics, and increase laboratory productivity. Through their premier brands, Thermo Scientific, Applied Biosystems, Invitrogen, Fisher Scientific, and Unity Lab Services, they offer an unmatched combination of innovative technologies, purchasing convenience, and comprehensive support. For more information, please visit www.thermofisher.com. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left-hand corner of your webpage and follow the process of obtaining your credits. I would now like to introduce today's speaker, Dr. Nicholas Dolman. Nicholas Dolman is a Senior Staff Scientist in Research and Development, Biosciences Division at Thermo Fisher Scientific. Dr. Dolman received his PhD from the Department of Physiology at the University of Liverpool and completed a postdoctoral fellowship at the NIH. Dr. Dolman has been with Thermo Fisher Scientific for 7 years. He has led the development of over 50 products that cover a diverse range of technology platforms, including fluorescent protein-based biosensors, Invitrogen, ClickIT, chemistry, and environmentally sensitive dyes. I will now turn it over to Dr. Nicholas Dolman for his presentation.
Thank you, Marjorie, and thank you all for joining us today for this webinar, where we'll go through high-content screening and its uses in life sciences research. So today in the presentations, we'll just start off a little bit by looking at some of the advantages of high-content imaging and analysis. We'll also look at what you need to actually perform the assays. What are the components that will allow you to perform a successful assay? We'll talk a little bit about the readouts. What are some of the biologies that you can easily address? And we'll touch on some of our bio applications contained in the Studio software. We'll also look at models. We traditionally think of high-content imaging and analysis as being for cell-based assays, but I'll show you data of its application as we move forward into different models as well. And then finally, we'll just touch on some real-world applications. We'll look at the use of high-content screening in cell health to give your cells a little bit of a sort of health check. How are they doing? Are they alive or dead? What are some of the cytoprotective mechanisms that may have been activated? We'll look at assays for proliferation as well using our novel ClickIT technology. We'll look at the application of high-content screening in actually making cell models. Here, we'll look at the use of stem cells to make neurons to model diseases. We'll also look at high-content screening and antibody-drug conjugates as well. How can we use this innovative approach to assess how effective a certain ADC may be? We'll also look at profiling when we've carried out a CRISPR genome editing of cells. How can we use high-content imaging and analysis and the broader range of reagents available from Thermo Fisher to find out what's happened to your cells following a CRISPR edit. And then finally, we'll also look at some screening applications where we've looked at a large siRNA library and some very distal cellular readouts. So what are the advantages of high-content screening? Well, there are a number of them, but let's remember that high-content screening is no different from standard fluorescence microscopy. You're still using a similar sort of optical path. However, with high content, we actually automate both the acquisition on the front end as well as the analysis on the back end. So we're still taking fluorescence images, okay? We're still using the same readouts that are familiar to the standard microscopy user. However, we're using the software to then automate and acquire images objectively. We're not, as the user, picking which cells that will be sampled. And then, of course, because the analysis is also automated, we can go through and very easily analyze a lot of cells. We're talking millions of cells within these plates. And how this does it is all within our algorithms contained in the Studio software. We're able to identify cells. Often this will be nuclear staining. We'll segment off the nuclei. So dyes such as DAPI or Hertz 33342, commonly used. There are other mechanisms to segment cells, of course. We can use whole cell stain, some of our HCS masks, or indeed cell tracker dyes for live cell applications. This allows the software to identify cells as it's acquiring, and then this is the basis for all the subsequent downstream analysis. Once we know where a cell is, we can start to look for properties within the cell. Here we see a field of DAPI-labeled nuclei, and in green here, this is clicked-edu with Alexafluor 488. So we know these cells are proliferating. The software is able to pick that up. We're sampling a huge number of cells, and we can plot any number of features that the software automatically acquires and start to look for patterns. We can look to gate on certain populations. We can say any cell with an intensity above or below a certain value we see as a percent responder or not. We can start to gate these, plot them, look at them, and because of the properties of the imaging, we're actually able to then correlate the data, the numerical values, with actual imagery. You can click on any cell in the analysis software at any data point and bring up an image of the cell. That's incredibly important for inline QC. Make sure your assay is segmented properly, that what you're reading out really is true and you're not picking up artifacts. And this all of our data comes through on the fly for analysis as you run. So let's just give you a quick example, an application very close to my heart. This is autophagy, and so what one would do coming into the bioapplication software is to click on the autophagy assay icon. This gives you preset settings for this assay. Here we're using a lung cancer cell line, A549 cells. We're going to label the nuclei with a cell-permeable DNA dye, Hertz 33342. Following drug treatment, we're actually going to fix the cells and immunolabel with an antibody against LC3B, the gold standard for autophagosomes. This is a very commonly used marker to show the presence or indeed absence of autophagosomes within the cell. And then we'll detect that with a secondary antibody, of course, using our premier Alexafluor 488 dye. So in these fields within the wells, we're labeling the cells with hooks. Now we're able to pick up the nuclei. Our software will say how many nuclei we have in a field, and we can ask the software to acquire a certain number of cells, and we generally do around about 1,000 per condition. We then take an image in the second channel, so standard microscopy approach. Here we're picking up the signal from the Alexafluor 488, which is reporting the presence or absence of autophagosomes. This is in the maximum drug treatment, so we're seeing an awful lot of autophagosomes present. We then build a ring. This is our cytoplasmic area. Here we've chosen just to take a relatively small region around the nuclei. That's then overlaid onto the image of the LC3B, and from there we ask the software to identify spots. So this is an automated spot counting algorithm. We can see here in the top, this is the minimal treatment vehicle. You see very few spots. The bottom is the maximum, which I showed you previously, and you can instantly see the difference in the number of spots. If you were doing traditional fluorescence microscopy, you'd either have to count these manually or import them into third-party software. Our packages do this automatically. This is a dose-response curve. This is 7 concentrations of the vacuolar ATPase inhibitor baphlomycin A1. It alkalizes lysosomes, inhibiting autophagic flux, and so we see accumulation of autophagosomes within the cell. There are a number of different readouts that we can take, and this is incredibly important to remember. Here we can look at ring spot counts, the number of spots present in that cytoplasmic ring. We can also look at the area and indeed the intensity of the LC3B staining. And these aren't criteria that we pre-selected. This all comes through automatically, and so you're actually able to mine your data sets, look for novel changing parameters. It's a very useful tool to interrogate novel biology. So what we're doing here, we have replicates of 6 with 7 concentrations of the drug. We're plotting the means from each well. This is a very nice way of getting reproducible and robust data. But what we're also able to do is actually mine and pull the data from every single cell within these wells. I'm just going to show you how we do this for ring spot area. Here, this is the data for the ring spot area from every single cell within all of those wells. There's several thousand cells, and this is a really important aspect of cell biology that high-content imaging is able to address, namely the heterogeneity within a population. Cells do not respond as one, they show differential responses. You can still see that dose-dependent increase in the ring spot area when we plot the data from every single cell. However, you can see that even at lower concentrations, some are responding and having a fair ring spot area. And at the highest concentrations, about 1 micromolar, we can still see some cells that aren't responding very much at all. So again, this is the heterogeneity, that spread of responders. We can also present it as box plots. This is the 25th and 75th percentile responders. And so there's all sorts of different ways that you can then take that data and represent it to really show that heterogeneous response. This is just some examples from our data using autophagy. I'd also point you to this paper in the literature from Lance Taylor's laboratory. Here they show and describe further analysis and acquisition protocols to look at the heterogeneity within cell populations. And in this published example, they look at phosphorylated STAT3 and show some beautiful examples of how cells show differential responses to reference compounds. So what do we need to carry out the successful high-content assay? Well, there are a number of things that you'll need to have in place to get this sort of data. The first is your label. So, for example, our reagents from Thermo Fisher Scientific. These will allow you to label cells and pick up a number of different responses, everything from apoptosis to autophagy, mitochondrial health, and so on. You'll also need the hardware. So these are things like our HCS instrumentation, and we offer a range of solutions from our benchtop Cell Insight CX5 right through to our newly developed Cell Insight CX7. The Cell Insight CX7 has inbuilt confocal capabilities, allowing you to optically section through thick samples, as well as laser autofocus to complement our standard image-based autofocus. And then, of course, we've had our flagship product, the Array Scan, which is also compatible with confocal acquisition, as well as liquid handling. And then finally, the instruments and the reagents themselves are nothing without that bioapplication software. And this is our newly developed HCS Studio 3 that contains newly updated bioapplications and all the analysis software to automate these highly complex fluorescence assays and make them really very simple to perform. So when we start to look at how these translate into life sciences research, we should look at some of the outputs. A very simple application is translocation. Here we show an example of nuclear to cytoplasmic translocation using NF-kappa B. We're able to look at the intensity from that ring region representing the cytoplasm, as well as the nuclei. And here you can see the software segmenting the cells. We take the nuclei again to tell us where we have a cell. We then use that to bring out and build up a picture of the cytoplasm as well as the nucleus. And here the compound is TNF-alpha, and we can see that robust translocation. Each of these colored traces represents the data from one row in a plate, so this is the replicates of 6 across a 10-point dose response curve of TNF-alpha. We can look at other readouts, not just intensity within regions. We can segment very fine structures such as elements of the cytoskeleton, microtubules, or the acrotin microfilaments. And here we have an example where cells have been labeled with Alexa 488 phalloidin. We can treat them with either a high dose, 10 micromolar of cytoclasine D to disrupt the actin microfilaments, or a much lower dose, which doesn't have an appreciable effect on those microtubules. The software is then able to identify the microfilaments, segment them, and we can pull off a number of fiber properties. In green, we can look at the number of fiber counts. This is how many fibers within a cell are actually picked up. We can also look at intensity in red. You can see the intensity increases, and that's evident from those images. What we see is as cytoclasine D disrupts those microfilaments, they condense down to points within a cell, which label very intensely. I showed you examples previously with autophagy and LC3B, but these same spot algorithms can be applied for other features. So, for example, here we're looking at the interaction between a ligand, EGF, and its receptor. Here we have a GFP stable cell line expressing EGF-GFP. We've labeled the cells again with Hertz to delineate the nuclei. Now we're using a fluorogenic EGF ligand. Frodo is a pH-sensitive dye that is essentially non-fluorescent in neutral environments. Only when this dye is internalized into endosomes and subsequently trafficked to the lysosomes do you see this dye light up. And so when attached to EGF, we can do these assays in a no-wash formulation. So we can bathe the cells in EGF. We won't see any signal from outside, only the EGF being internalized. We're going to modulate the internalization in this assay using an inhibitor of clathrin-coated mediated endocytosis, a compound called pitstop2. At low concentrations, we can see that there are spots forming within the cells that are positive for Frodo-Red EGF. If we use higher concentrations of pitstop, a very short pulse of 30 minutes, we can see complete inhibition of the internalization of both the receptor as well as the ligand EGF. And again, we can plot this as a dose-dependent readout. Here we see the dose-dependent decrease in receptor internalization, mirrored by dose-dependent inhibition of ligand internalization as well. And finally, another example where, rather than using intensity to look at translocation, we're just looking at the intensity within a specific compartment, reading out a biological response. Here, DNA damage, using an antibody against the DNA damage marker, phosphorylated H2AX. We're treating cells with the etoposide and looking for the single intensity from this antibody. And you can see as we go up in concentration, we see a dose-dependent increase in the intensity of our antibody labeling. And these are just a snapshot of all the different biologies that can be read out, and we'll show you a few more as we go on through the presentation. So what about models? Well, I alluded to this earlier. High-content imaging and analysis isn't just for your cell-based assays. It can be applied up to 3D structures as well as model organisms. A very useful tool is the use of 3D spheroids. Here we have a kidney spheroid labeled with a confocal unit on the cell inside CX7. And we can see active and high-fidelity segmentation of nuclei within this 3D spheroid. And because we're able to optically section with our confocal capabilities and build a 3D representation of these spheroids, we're able to do more complex assays. This is a HeLa spheroid. We're using our live dead assay. Live cells are green, dead cells are red, and you can see the increase in the number of dead cells as reported by the red indicator when we switch from vehicle to a cytotoxic agent, niclosamide, in a 3D culture. We can also look at other more physiological functions. Here is an MCF7 tumor spheroid. We've labeled with Hurst, again to tell us the number of cells within the spheroid. Now we've labeled them with our newly developed hypoxia reagent, the Imaged Hypoxia probe. This sensor lights up or gives rise to fluorescence when it's in environments of reduced oxygen or hypoxic conditions. So here we're able to image the hypoxic core within a tumor spheroid. And of course, much like our confocal capabilities enable spheroid research, they also help in other thick tissue or samples. Here we have an embryoid body. We're taking a maximum projection of 20 slices, comparing the performance with confocal on the right versus wide field when we look at the anti-beta-3 tubulin staining of this embryoid body. We can also image tissue. Here's a kidney tissue using some Alexaflor 488 wheat germ agglutinin. And you can see the improvement in resolution with our cell insight CX7 confocal versus wide field. And finally, we can apply this to complex model organisms. Here is an image and overlay the segmentation of a zebrafish. This is an angiogenesis model using the zebrafish as an organism to study angiogenesis. And now we can look at some vessel properties, obviously highly important to angiogenesis and its study. We can look at the vessel count and vessel length as a function of various concentrations of two compounds. So we're able to apply high-content imaging and analysis not just from cells through to spheroids, to tissue, and into model organisms. We have data on other model organisms as well, things like C. elegans and the like. So let's take a look at some applications in real-world examples, and I'll just list those out for you too. First, to start, we'll take a look at cell health. So we can think of the health of a cell as a continuum. They can go from healthy to signs that all is not well, a sort of a sign that the cell is unhealthy. We can also look at cytoprotective mechanisms. These are activated to take the cell back from not being particularly healthy back to a well state. We can then look at markers at the point of no return, after which the cells cannot recover and they progress to die. As we look at the induction of cell death, we can look at the mechanisms, such as apoptosis. We can also read out whether or not the cell is dead. We're also developing tools to look at some of the consequences, the cleanup, as it were. How do cells of the immune system deal with dead cells and translate these into cell-based assays? The health of the cell is incredibly important, and it's an often studied area because really, as researchers, we often fall into one of two categories. We're either interested in trying to keep cells alive, so if we work in post-mitotic cell types such as neurons or cardiac myocytes, these can't be replaced, and so it's incredibly important to try and promote their survival. Of course, if we work in oncology or cancer, we're on the opposite end of the spectrum. We're looking to drive cells to die, so we're looking at indicators that cells are dead. And really, almost everybody in the basic research field falls into one of two camps. So we spend an awful lot of our resources developing assays and showcasing their use of high content to read out these various portions. And there are really a number of different ways in which you can look at any one number of these points. Viability can be really any function of normal physiological actions within the cell. These are some of the hallmark ones that we look for. Controlled protein synthesis and RNA synthesis, polarized mitochondria, which will produce ATP. They also won't be giving out ROS bursts, will have a predominantly reducing cytoplasmic environment, as well as regulated proliferation. Some of those signs that all is not well, or pre-lethal indicators, the kind of canary in the coal mine, as it were, a loss of protein quality control, depolarized or depolarizing mitochondria, an oxidizing cytoplasmic environment, which is often underscored by the production of ROS bursts, lower ATP concentrations within the cytoplasm, as well as deregulated proliferation. When we start to think of some of the cytoprotective mechanisms, we're thinking about things such as autophagy or DNA repair. When we think of mechanisms of cell death, we're talking about apoptosis or necrosis, or its program form necroptosis. And in viability, almost always we look at a loss of the plasma membrane integrity. And then we can read out some of these immunological consequences by looking at assays for phagocytosis, which will report these find me, eat me signals, as well as various aspects of the inflammatory response, things such as these danger-associated molecular patterns, which signal whether or not an inflammatory response will be initiated. Today, just for reasons of time, I'm just going to show you a few of our probes, their application in high content for these three areas: loss of membrane integrity, caspase 3-7 activation, as well as polarized mitochondria. So as I said before, one of the most common ways of looking at viability is through plasma membrane integrity. But we can actually add an additional level of reporting here by looking at enzyme activity. Living cells will often have certain key enzymes active, and so by labeling them with reporters of this enzyme activity, we can report not just death, but as well as live cells. We'll typically use a green dye, such as calcium AM, to report the live cell, and we'll use an impermeant red nucleic acid binding dye. These can't enter the cells, and so green cells will show up, live cells will show up green. They will not have any red signal. Upon induction of cell death, we see a loss of plasma membrane integrity. This means that the green dye is no longer retained. It leaks out, you lose the green signal, and you gain a red signal. This is what it looks like when we treat cells with a low dose of a cytotoxic agent, just enough to kill a few of the cells. We'll see a mixed population of live cells in green and dead cells in red. We can read this out in high-content imaging analysis using intensity or percent responders. Here's an intensity readout. We're looking at the intensity from both the green and the red signal. We see a drop in the green signal with a concurrent increase in the red signal as a function of increasing camptothecin concentrations, camptothecin being, of course, highly cytotoxic agent, which will very effectively kill cells. We can also look at the mechanisms in which a cell employs to activate cell death. Here we're going to look at the activation of caspase 3-7. Caspase 3-7 is activated both through the intrinsic and the extrinsic pathway, so it's a good readout for general apoptosis. We have a fluorogenic reporter, cell event caspase 3-7, and we can see when compared to untreated cells, cells treated with etoposide show a significant increase in the intensity of this label. And indeed, this change, this time not looking at intensity, but percent responders. So we ask the software to gate that over a certain intensity value, we'll say that they are apoptotic. Now we look at an increase in the number of cells undergoing apoptosis as a function of compound treatment. Here, staurosporin, a broad-acting kinase inhibitor, which induces apoptosis. We see a dose-dependent increase in the signal from this reporter. It is specific for apoptosis. We can keep apoptosis at a tonic level with a low dose of staurosporin and apply increasing concentrations of a caspase 3-7 inhibitor. We see a dose-dependent loss of the signal, reflecting its specificity to caspase 3-7. We can also back this up with genetic models. Here's a CRISPR-Cas9 model where we've knocked out caspase 9, and we can see no signal in the caspase 9 knockout with increasing concentrations of staurosporin, yet we see a dose-dependent increase in the matched parental line. And of course, caspase 9 is just upstream of caspase 3-7, so it's a very nice specific modulator of caspase 3-7 activity. There are a lot of ways of looking at mitochondrial function, specifically mitochondrial membrane potential. Here I'm just going to showcase one of our reagents, TMRM, or tetramethyl rhodamine methyl ester. It's a potentiometric probe that accumulates within polarized mitochondria. We can see that it is truly mitochondrial as it overlaps with our Bachmann cell-like mito-GFP, so we are getting very good mitochondrial loading. We can look at the intensity from these mitochondria as a function of protonophore concentration. So protonophores such as CCCP will depolarize mitochondria. And here's a high-content assay looking at 1 hour after compound addition in 3 different cell lines. These are replicates of 6,000 cells per condition. So you can see here we're doing a very, very high number of cells across a number of cell lines. We're able to do it very, very rapidly and efficiently. Of course, we can multiplex these reagents and start to look at the sequential induction of apoptosis. So here we can look at the loss of mitochondrial membrane potential and subsequent activation of caspases for apoptosis using TMRM and cell event probe together. We can then build this a step further by using some of our mitochondrial morphology probes, such as the Mitotracker probes. Here we use Mitotracker deep red, TMRM, cell event caspase 3-7, and of course TERP to segment the cells. Now we're able to look at mitochondrial fragmentation, the loss of the membrane potential, and subsequent activation of caspases. So assays for proliferation are very, very popular. We've developed the CLICKIT-EDU assay. So this is a BRDU type assay. Rather than looking at the incorporation of BRDU, when cells are in S phase, we use a different thymidine analog, here a thionyl diuridine. It's CLICK compatible, so we can do this very nice bioorthogonal chemistry to link a reporter, a zeta-modified alexafluor dyes, onto that EDU. So cells are fed EDU. If they're proliferating, they will incorporate them into the nuclei. We then fix and permeabilize, carry out the CLICK reaction, and now we're able to very precisely label them. This is a culture of human dermal fibroblasts. We pseudocolored the alexafluor 88 CLICKIT-EDU in orange. The total number of nuclei are in purple, and then the microtubules in cyan. And you can see that within this 3D culture of human skin cells, we can see quite a few cells that are actively proliferating, as reported by the incorporation of EDU during this short 30-minute pulse. We can also look at this, use this tool to look at changes in proliferation. Here we're using RNAi to knock out a molecular motor involved in mitotic exit. If we knock this motor down and wait for 48 hours and then do a short pulse of EDU, we see that cells aren't exiting mitosis because of the loss of kinesin 11. They're not free to then enter S phase, so you see a reduction in the EDU incorporation in this population of cells. So a very nice tool for looking at modulation of proliferation. Not just genetically, but also chemically. Here we have a dose response of the commonly used mitotic inhibitor Taxol, and we see a very nice dose-dependent inhibition of EDU incorporation in this assay. So EDU is very useful for looking at proliferation, but it can actually tell you other aspects of cell biology. We can combine it with DNA dyes to look at DNA content, antibodies against specific markers of the cell cycle. So here we're using DAPI for DNA content, tell us the 2N and 4N population. We can use these S phase markers to tell us which cells are in S phase. And then here we're using phosphorylated histone 3 as a G2M marker. And we can build these almost flow cytometry-like plots of cell cycle progression. We can start to say which cells are in G1, which are in S phase, which are in G2, and which are potentially blocked in G1S. And again, we're sampling a large number of cells, so we're getting very robust and reliable data sets. We can also use proliferation as a very sensitive marker of prelethal toxicity. So this time we're going to pair an impermeable nucleic acid dye, image it dead green, with our Clickit EDU Alexa 647 assay. So we're using the FITSI and Sci5 channels. So live cells will have a certain level of proliferation as reported by red nuclei, but they won't have any report from the green dye because they haven't died. If a cell was to die, the green dye would gain access. Of course, once the cell's dead, it's not proliferating, so it won't incorporate EDU. We can actually use that to look at changes that occur before death is even induced. Here we're using a very high concentration of camptothecin, but a very, very short pulse, only a few minutes. What you can see is there really isn't a large induction of death, as reported by a dead green. However, what we see is a very prominent shutdown of S phase, the cell cycle. The cells have sensed something's wrong. They haven't died yet, but the loss of proliferative index in this cell population is an indicator that all is not well. So it's another way in which we can look at prelethal toxicity in a multiparametric fashion using spatial readouts amenable to high-content screening. So now we'll actually use high-content imaging and analysis to set up some biological models. We're going to look at neuronal differentiation. Here we have a model from our colleagues in GIBCO where it's the production of dopaminergic neurons from stem cells. Starting with these human pluripotent stem cells, the kit actually allows you to differentiate into the fluorescent progenitor, or FPP cells, which are the intermediate step before production of mature dopaminergic neurons. These are an excellent model for various human diseases that can be studied for basic research. And we actually have a very robust workflow that allows you to produce these mature neurons within about three or so weeks. And of course, high-content imaging and analysis is a very useful tool to deploy alongside this protocol. What we can do is we can look for markers that these fluorescent progenitor cells have been formed. So the presence of FOXA2 and OX2 tell us that we have these fluorescent progenitors. We come in and we use that segmentation and target activation module to tell us that we have these molecules present. That way we can segment, say how many of our human pluripotent stem cells have transferred into this intermediate state. From there, we then mature them into mature dopaminergic neurons, and using TH, which is tyrosine hydroxylase, as a marker of mature dopaminergic neurons, we're able to read out how successful this protocol is. We can segment, look for the presence or absence of this marker, count the number of nuclei, count the number of cell bodies showing positive for tyrosine hydroxylase, and from there calculate how many of these cells are positive and are indeed transformed into mature dopaminergic neurons. And of course, high-content screening doesn't have to stop there. We can actually look at more detailed neuronal properties, for example, synaptogenesis. So what we do here, we're still employing a nucleic acid stain, DAPI here. We're then going to label those neurons with a neuronal marker, MAP2. Then using antibodies against both pre- and postsynaptic structures, synaptophysin for presynaptic regions and PSD95, postsynaptic density 95, the postsynaptic marker, we're able to look at synaptogenesis. Our software is able to segment the nuclei, from there build up a picture of the cell body. Once the cell body has been identified, then the projections, the axons and the dendrites can be segmented, and from that we can overlay spot analysis of those pre- and postsynaptic markers and quantify how many synapses are present within this neuronal population. And we can quantify that and indeed look at the effect of adding certain compounds, so kainate and glutamate, which will modify and modulate glutamate receptors. So here we're just looking at the number of pre- and postsynaptic vesicles, and from that reading out the number of synapses. But we can also look at other properties of these neurons that may correlate with the compound treatment. We can look at the number of neurons and neurites. We can look at the intensity of staining, their total length. We can do branch point analysis, neurite width. So there's a lot of different aspects of neuronal biology that come through automatically and then can be mined. We can also use this same advanced segmentation of neuronal cells to look at toxicity. So if we look at stimulation of control cells with NGF into more mature neurons and then subsequent cell death assays, for example with cytotoxic compounds such as retinoic acid, we can look at segmenting the neurons as a readout of how healthy they may be. We can look at the number of cells, the neurite branching, the number of neurites, as well as their length, and actually start to plot how these various features change as a function of compound treatment. We can also assess the effectiveness of an antibody-drug conjugate. So antibody-drug conjugates will bind to a cell surface receptor. They'll often be internalized, traffic to the lysosome. There, the lytic enzymes activate a compound. They then diffuse out of the lysosome via permeases and act on their cytoplasmic target. We're going to show you a sort of complete assay here. Here we've labeled a therapeutic antibody, Herceptin, using our new site-click chemistry. This is a site-specific and defined conjugation approach. Here we are able to label them and just look at the uptake of these antibodies. So we're using SKBR cells and MDA-MB231s. SKBR cells are HER2 positive, so Herceptin is able to bind. MDA cells are HER2 negative, so Herceptin won't bind. We can use an unrelated therapeutic antibody, Gazyva, a relative of Rituxan, which won't bind to HER2. What you can see from this trace, we're just looking at the membrane intensity using high-content imaging and analysis. You can see we see a significantly larger signal from the S-dibo, which is our click chemistry method of labeling these antibodies with Herceptin, and in no other situation do we see that signal. So there's no signal when we label Gazyva using click chemistry on HER2 cells, HER2 positive SKBR cells. And of course, we see no signal from either Gazyva or Herceptin in HER2 negative MDA and B cells. So not only is Herceptin binding specifically, but these therapeutic antibodies aren't able to be just taken up by a fluid phase endocytosis. We can look at the time and dose response here. So these are SKBR cells, so HER2 positive. We can label them for 29 hours or 9 hours or 5 hours and look at both the time and dose dependency of the uptake as well. We can then modulate this click labeling approach and actually add on the toxin as well as a reporter. So here we label the antibody not only with a reporter, Alexa 647, but also with a toxin. In this case, it's MMAE, a microtubule inhibitor. These depolymerizes microtubules, triggers mitotic arrest, which then subsequently causes the induction of apoptosis. We use our model, as I described previously, of SKBR3 cells for HER2 positive, as well as the HER2 negative MDA line. You can see there's no signal from the Alexa 647 in the MDA cells, but we do see a very bright and robust signal for the SKBRs. We can look at the uptake of Alexa 647 into these SKBR cells, and then we can also overlay that in the same cells in a multiplex experiment with the induction of apoptosis using cell event caspase 3-7. And of course we can look in the HER2 negative MDA cells, and we see not only is there no uptake, there's also no induction of death. Look at some screening applications. We're just going to show you an example of an siRNA library. Here we used a small focused library against the kinome, so that's all the kinases in the genome. We use cytoskeletal analysis as a distal point, so looking at how these kinases across the spectrum may affect cytoskeletal structure. We were looking not only at F-actin, as I detailed before, but also microtubules. We'll also use some of our the whole cell stains, the cell masks, to look at cell and nuclear morphology to see if they're changing. Software segments these cells, is able to identify both actin microfilaments as well as microtubules, and from there perform a high throughput quantitative analysis. We're able to look at 52 cellular properties simultaneously across the treatment of various kinases knocked down with siRNA, and we can cluster them based on conserved responses across those 52 parameters. We can then cluster them. We'll visualize them along the two main component analysis axes and start to group them and have a look for how these genes may fall into conserved signaling pathways across the board. We can actually look at which kinases fall into which clusters. So finally, I'll just end up and show you an example of using high-content imaging and analysis in combination with Thermo Fisher fluorescent reagents to perform phenotypic profiling of CRISPR-Cas9 edited cells. So why did we do that? Well, obviously CRISPR-Cas9 is a very popular system. It's been in the news a lot recently, and it's a lot of researchers are looking to incorporate this workflow into their research. When you perform a CRISPR-Cas9 edit, one of the confirmation experiments you'll do is maybe sequence them to confirm that you've performed the knockout successfully. You may also do a GCD assay, so genomic cleavage detection. So this will also confirm that you've got specific nuclease activity at the loci of interest. Of course, whenever you're knocking out a gene, it doesn't have to be with CRISPR-Cas9, it could also be with RNAi, it's critically important to check that the protein has gone. Because if your protein has an extremely long half-life, these two references here discuss some of the importance of protein half-life to cells. It doesn't matter if you've lost the gene, because the protein will still be there to act. But we're going to show you why it's so important to also do complementary cell-based assays, because although you may have lost the protein, there can be occasions where other proteins can compensate and kick in, so now you're actually able to confirm the phenotype that you'd expect with that genotype. I'm just going to show you a very quick example to start with of a phenotypic assay. Here we're looking at knockout using CRISPR-Cas9 of GCLM, the glutamate systase ligase moiety. It's involved in the synthesis of reduced glutathione, and reduced glutathione is one of the main cytoplasmic buffering mechanisms to protect cells against reactive oxygen species induction. We blot using an antibody against GCLM, and we can see that we've lost the protein. So what are the functional consequences? So we've knocked the gene out, and these genes have been sequenced as well, and we can confirm the gene's been lost, as has the protein. But now we can deploy a proximal assay, so very close to the gene of interest. This is file tracker violet. It's a direct measure in cells of the amount of reduced glutathione, and we can indeed see that the knockout cells show significantly reduced glutathione levels. So here our cellular assay, a phenotypic assay, has complemented our proteomic and genomic assays. And these are very important to do when you're knocking out a gene, can make sure that your cells are displaying the right phenotype. But we can take it a step further. We can actually use cell-based assays and functional assays to construct a complete picture of cellular function to look at the consequences of CRISPR-Cas9 editing. We can combine assays close to the gene as well as far away, so both proximal and distal assays. And I'm just going to use that example of autophagy again. Autophagy is a complex multi-step process. One of the most commonly deleted genes is ATG5. It's involved in the conjugation of LC3 to the autophagosomal membrane. Without ATG5, LC3b cannot be conjugated to phosphatidylethanolamin. It won't associate with autophagosomal membranes, and the cells are deficient in macroautophagy. Here we have a Cas9 stable cell. We've transfected these cells using our neon electroporation system with guide RNAs against ATG5. When compared to the parental or scrambled transfection, the ATG5 transfected cells show a significant loss of LC3b spot intensity as a function of chloroquine. Chloroquine acts in a similar manner to baphlomycin to block autophagic flux. These are pooled cells, so not all of them have successful knockout. If we then go and take this pool and select clones, we can blot and show that ATG5 was lost in these 5 clones. We then pick a clone, clone 3. We do subsequent analysis. Here we're actually using co-localization analysis. One thing that you'll see when you block autophagic flux is the co-localization of LC3b for autophagosomes with lysosomal markers, and here we're using an antibody against LAMP1, a lysosomal resident enzyme. You can see in the parental line in red, we see increasing co-localization of these 2 markers as a function of chloroquine concentration, whereas in the knockout line, clone 3 of the ATG5 knockouts, we see no overlap, we see no formation of LC3b positive puncta. This cell is deficient in the formation of autophagosomes. It won't be able to carry out macroautophagy. So we've confirmed with a proximal assay that indeed the cells display the phenotype that would be indicated by both the proteomic and genomic studies. But we can then take it a step further and start to look at related processes. P62 or SQSTM1 is a receptor for targeted autophagy. It recognizes ubiquitinated cargo and binds to that cargo. It also binds to LC3b, thus bringing that cargo into the autophagosome. We can look at the number of spots in the wild type or the parental line versus the knockout, and what we see is, as expected, the number of P62 positive spots increases when we block autophagic flux in the parental line. However, the knockout line shows us a similar increase in the number of spots, and actually also subsequently increases when we treat with chloroquine. This is what we'd expect. The knockout line are unable to carry out macroautophagy, and so we won't be able to clear P62. However, it was intriguing to see true puncta forming. We weren't really expecting that, so we could start to look at where they are. We can blot and show that there are indeed increased levels of P62 within these cells, However, these puncta aren't getting into the lysosome because in live cells, using our Bachmann primo autophagy GFP-P62, we see that they don't overlap with the lysosomal marker, lysotracker deep red. Chloroquine-treated wild-type cells, those P62 puncta do overlap with lysotracker deep red, showing that they've been correctly trafficked to the lysosome. They just can't then be degraded. However, in the ATG5 knockout, those puncta do not co-localize with the lysosome, indicating that they're accumulating within the cytoplasm, which would be consistent with the elevated levels of P62 in these knockout cells. So because autophagy is primarily a pro-survival mechanism, it helps cells to withstand periods of nutrient deprivation, it clears damaged organelles, as well as helping to deal with invading microorganisms and so-called xenophages. We wondered how healthy these cells were. During our cell culture, we'll assess viability with our cell counter, and we actually noticed there was no difference in viability. So maybe the cells were still alive, but perhaps they were teetering on the edge and were ready to die at the slightest stress. Well, to do that, we looked at the induction of apoptosis, the same model as I showed you before. This is using cell event caspase 3-7 and staurosporin to induce autophagy. And we see no significant difference in the induction of autophagy between the parental line and the ATG5 knockout. So not only are these cells healthy in culture, they're also not predisposed to cell death by the induction of apoptosis. However, staurosporin and kinase inhibition may not typically target autophagy. So we wanted to start to look at maybe more autophagy-related processes. So we looked at the fusion, so because autophagosomes fuse with the lysosome, we started to look at some of the consequences of ATG5 knockout with the lysosome. We used a number of different tools for lysosomal demarcation. So we used anti-LAMP2, and we saw no significant difference in the number of LAMP-positive vesicles between the parental and the knockout line. When we did live assays using lysotracker, so a lysomadropic compound, we actually saw a significant increase in the number or indeed the area of these lysotracker positive spots, suggesting some dysfunction in lysosomal properties between the ATG5 knockout and parental line. Even though there don't seem to be any difference in the number, there is something different in terms of their functional properties. We then used a second orthogonal readout of lysosomal activity. This is a BODI-PFL fluorescent conjugate to the drug pepstatin A. Pepstatin A inhibits cathepsin, binds to active cathepsin, so we can use it to read out cathepsin activity within cells. And we see a significant increase in the BODI-PFL pepstatin labeling in the knockouts than compared to the parental cells, showing these cells do have significantly altered lysosomal properties. Because autophagy is involved in organelle homeostasis, we're intrigued to see what happened to other organelle function. Here we used Mitotracker Orange to look at mitochondrial properties, and we saw there was actually a decrease in the number of mitochondria in the knockout compared to the parental cell line. However, when we looked at the area occupied by the mitochondrial labels, it was actually significantly higher, suggesting some dysfunction in the balance between fission and fusion of mitochondria in the ATG5 knockout. We could also look at functional properties. So although both pools of mitochondria in the knockout and the wild type were able to be depolarized by CCCP, it appeared there was a significantly higher polarized state of these mitochondria. They showed increased TMRM intensity in the knockout compared to parental line, again suggesting some difference in mitochondrial function. So we actually took this a step further and started to look at other organelles. We could look at the area occupied by a Golgi label. This is using anti-Golgin 97. We see no significant difference in the area occupied by Golgi labels within the cells. We do, however, see a significant reduction in the area occupied by the endoplasmic reticulum. Here we use a marker against phosphodiesterase isomerase, PDI. We see a significant reduction in the ER area of the knockout cells. We also see some perturbation of actin cytoskeleton function. We see slightly higher numbers of fibers within ATG5 knockout versus parental. And finally, we're able to look at peroxisomal biology. So peroxisomes are specifically cleared via a process known as pexophagy. And in these knockout cells, we see a significant reduction in the area occupied by anti-PMP70 labeling. It's a peroxisomal membrane protein. So using high-content screening, we're actually able to look at not only processes close to the gene of interest, ATG5, at autophagy or its clearance of its receptors, but start to probe some of the more distal and potentially unrelated processes we're actually seeing are affected by knockout of this gene. So things like organelle mass, organelle function. So again, it just highlights the broad number of uses of high-content screening in the life sciences research. And with that, I will finish up for today. I hope you found today's webinar useful. We will be taking some questions. We may not have time to get round to all of them right now, but if we don't, we'll be following up after the event. So thank you very much.
Thank you for that informative presentation. Before we get started on the question and answer session, I would like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, how accurate are these techniques using 3D tumor model are tested for spheroids or also for the hydrogel-based model? Can you talk a little bit more about how high-content imaging is being used today to look at organoids and 3D models? For example, what measurement systems or markers are they using?
So yeah, that's a very good question. Not only do you need the optical systems that are able to image within these spheroids, things like very, very robust light sources from the LEDs, but also confocal capabilities for detection. You do also need to validate the reagents, make sure they're permeating all the way into the spheroid. So we are actively working on validating a number of our reagents in these models, looking at ways in which we can improve permeability, reduce non-specific binding as well.
Thank you. And our next question is, we often find the center of microtissue dark with confocals. Is this due to the opacity or due to the stains not penetrating into the center of the tissue?
It's almost certainly a combination of both of those. So you could be having an issue of the light getting in to excite the fluorophores, the light then exiting and not being scattered, as well as the penetration of the probe.
The next question is, what kind of coating plates for neutron culture?
Sorry, could you repeat that? I missed the last bit.
What kind of coating plates for neutron culture?
I don't have any experience in that area, but what we could do is actually follow up offline on that. We can put you in touch with some of our plate teams.
How fast or quickly can a single fluorescence image be acquired for a typical imaging application?
It will vary from application to application. I generally don't have acquisition times longer than 100 milliseconds. I would say typically from about 10 to 100 milliseconds for your standard fluorescent reagents and secondary antibodies. Of course, the Herx labeling is even lower, often a millisecond or two. Every now and again, you may have to go a little bit higher than 100 milliseconds, especially if you're using a very dim reporter. But generally speaking, about 10 to 100 milliseconds.
Okay, it looks like we have time for one more question. How long are the exposure times?
So similar to that last question, relatively speaking, about 10 to 100 milliseconds is what we aim for. It's rare that you need to go higher. Every now and again you will, but on almost all of those images you see there, on the low side of that, around about 50 milliseconds or so. And of course, the exposure time will affect your total acquisition time for a plate. So again, we always try and get very robust and also not only bright but stable light sources. You don't want your light source fluctuating in power because that can give you false differences in the intensity read out.
And the last question is, high-content screening is a relative new addition to the drug discovery process. Can you comment on how you have seen customer use HCS in a drug discovery campaign, as well as comment on some of the common hurdles or challenges people are running into?
Yeah, I mean, we've seen high content really deployed across the spectrum of drug discovery, not only from the standard screening of libraries of compounds, from hit to lead campaigns, but also in discovery biology, and pathway analysis. So you can actually start to look and interrogate the biological signaling cascades that might be targeted in a therapeutic campaign, as well as once you have your target in mind, actually screening whether or not the compounds are working. So it's really deployed across the board, as well as in toxicity profiling once a compound is a little further on. And again, for any questions we haven't been able to answer, we will get back to you and answer those. So we apologize if we weren't able to answer your questions because of time, but rest assured we will follow up with you.
Thank you, Dr. Dolman, for your presentation. I would also like to thank our sponsor, Thermo Fisher Scientific, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through December 2017. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time, and goodbye.